Three from Malaysia, from Infomed, and I initiated this training. This is for the first time we are going to do it for Malaysia. This is something very new, and I must thank Prof. Dr. Roberto and Radha from Dubai for agreeing to conduct this session, which is going to last probably 1.5 hours, 2 hours. I came across Full DNA during last year's conference, which we organized in Malaysia, called the International Selangor Healthcare Conference. And Dr. Roberto was there to present a paper and conduct a forum. And we realized that he has some product on genetic screening, which is becoming popular, which is gearing us towards personalized medicine, something very different, very clinical. And we decided to engage with him, and over the last few months through our process, we realized that it is something which we should take it on for Malaysia. And this will be the first session for doctors who are going to be counselors and clinicians to manage the reports with the patients who are going to go through this screening. I would also like to inform here that during this year's conference, which is an annual conference which I organized with the Selangor State, one of the days we are allocating for precision health. So you will hear more, and I would like to invite all of you to join us during the conference to get more in depth into precision health. Now, finally, I would like to welcome those who are joining us from Malaysia and outside the region for this wonderful session. And I'm sure after listening to Dr. Roberto over the last few months and having a session with him, this is going to be super interesting. And this is probably, we are talking about the future of healthcare, how it's going to be managed. So it's going to be an interesting session, and please stay on with us until we are done today. And this is not the first one. We will have a continuous training sessions for us to keep us updated. There are a lot of new things that's coming up. We should also listen to that application with using AI, which they have developed, Full DNA. Another interesting application to assist the doctors in managing these genetic screening reports. So with that, I would like to thank you again for spending your Friday, a good Friday with us. I hand over to Radha to take on from there.
Thank you. Thank you very much, Mohan. It's been a great effort from your side, and we're very happy to see all the amazing people who are joined us here. I'll quickly give the background of the company. The company was started in 2011, and we are currently operating in more than 12 countries. And we are very happy to announce that we've just launched our Southeast Asian operations. We got the company floated in Singapore, and we have started now to embrace the markets in Malaysia, Indonesia, and Singapore. So we're eagerly looking forward to your support and cooperation. And on the same note, we would like to communicate that we have been more clinically focused when the whole world was focusing on recreational genetics and ancestry. Our clinical focus was mainly because Dr. Roberto comes from a cardiology background, and he really wanted to make a difference from the genetic angle towards providing diagnostics and not just alarming the patients with the vulnerabilities, but also to provide solutions. What can be done is our priority in terms of this offering. It's not giving just a report. So this journey has been very wonderful, and we have done more than 2.2 million genetic tests across the 12 countries, and around 5,000 doctors are currently prescribing our panels in South America, in Europe, in Middle East, and also starting in Asia now. So we look forward to your great support, and we are always here to provide whatever clarifications or validations that you may require to start practicing this as part of your healthcare approach towards your patients, because the science have evolved so much, so great that we are able to have absolute clarity of the cause of all the expressions of the genetically driven diseases, and not just the causes, we are also having solutions to fix them. So we are very happy and eager to present what is currently scheduled for today. And as Mohan said, we will be doing it every month, one session for our regional markets. And wonderful to have you all. Dr. Roberto, I think you can start the presentation.
Thank you so much. I'm Dr. Roberto. I'm very happy to have you online. These sessions will talk about different issues in DNA area. First of all, I'm going to start sharing my screen and start our presentation. And after doing the presentation, we're going to have 20 minutes of questions and answers for everyone. Okay, so let's start first. Share my screen. Here we go. So this is the genetic test analysis and actionable diagnostic systems. Okay, genetic test we know for a long time already. It's becoming a kind of commodity. You can do in almost every hospital, in many clinics in the world, and even you can do online. You can buy genetic tests that we call recreational genetic tests to understand the genetics of your family, your ancestry. But we're talking totally different subject. We're talking about how genetic can be used to detect diseases, to work with the prevention of diseases, and what are the actionable diagnostic systems, and what you can do in order to treat patients after receiving a diagnostic test in genetics. Okay, so this is the first part. We're going to explain to you what we do and how important is the tool that we're just releasing, just launching this new tool we call ADS, which is the Actionable Diagnostic Systems. And you are one of the first doctors in the world to see this tool. Here. The first part is about the DNA structure. Okay, I like to start my presentations telling you about what we have here. We have our cells. You can see here. This is our cells. We have in our body around 30 trillion cells. 30 trillion cells. Yes, and all of our cells, they have the same structure. They have a nucleus, except, of course, the red blood cells, but we're talking about most of our cells. So they have the same information. Okay, they have inside a cell, the human cells, 23 pairs of chromosomes. Okay, when I talk about the chromosomes, as we can see here in this drawing, in the shape of X, if we open the chromosome and unroll the chromosome, we have the DNA strand. As you can see, the DNA strand. But the DNA strand is so much compacted, and they are compacted around some protein structures that we call histones. But they're so well rolled that we cannot see them. But if we open, for example, just one DNA from one chromosome, we have something like 1.7 or 175 meters high. Okay, 1 meter, 75 centimeters high, tall. But it's so much compressed and rolled that we cannot see it. Okay, so all the structure of the DNA are very similar. We have the nucleotide base pairs. We have the nucleotides that we call the guanine, cytosine, adenine, and thymine. So this is written since we are born. When we are born, we have this information here, and we barely change that. Okay, we can have changes in our DNA, but they are not so common after we are born. Okay, but many people ask me, Doc, is this a life sentence? I need to have this for all my life. So if I have written my genes that I will develop an Alzheimer's disease or a cancer or a cardiovascular disease, it's a verdict? No. Why? Because we have some very important regulatory functions. For example, the methylation process that we can see here that determines which genes are going to be turned on and turned off in our body. Okay, just to explain to you how it works, in other words, I just mentioned to you here in this drawing that all the cells, they have the same information. That means that our heart cells has the same information from our liver cells and even from our adipose cells. They have the same chromosomes inside. But why these cells, they know they should be a liver cell, they should be a heart cell, or even an adipose tissue? How would they know that? Because the genes are expressed more and less in different types of cells. And who determines this expression is our DNA methylation system. So this DNA methylation system is a whole process where methyl, which is a chemical compound, is donated and is connected to our cytosines from our DNA. Okay, so you can see on the left side, adenine, cytosine. So imagine you have an open position, and then you have an H3C connected to this position. That means the methyl is connected. When the RNA comes to do the reading, and as you know, the RNA is like a photocopy machine, comes and copies information that it finds in the DNA. It's not going to be able to read what is occupied. So if the cytosines are occupied, they are not reading those positions. So if they don't read those positions, the gene information is compromised. And that's what we call the gene is off. Okay? So to have the good regulation system in our DNA, in our bodies, we need to have a perfect methylation process. But it not always happen like that. Okay? Many factors, our foods, our environment, ways of life, can really change the methylation standards in our body. And that influence the way we develop diseases, influence the way we reach or not longevity. So this is another drawing showing a little bit of the chromosomes, the methyl groups being donated to the cytosines of our DNA, turning that genes, making that position not able to be read by our DNA, but by our RNA. This is a process that happens every single second in our body. In order to determine the information of our risks, we have more than 5,000 diseases, different diseases and conditions. And we divided these diseases in different panels, in different subjects of the medicine. So here are just 15, but we have more than 40 different panels, and they are the most complete available today. Why? Because we cover the largest quantity of genes analyzed, and we are updated according to the information given to the American Pathology College of Pathology. Is in a day 24/7 information. So we are the most accurate and updated genetic database in the whole world. Okay, what is so important? For example, we have in diabetes, we have in cancer, in longevity, cardiovascular, neurodegeneratives, and even in autism. We have information. How is your body turning on, turning off? How is your genes working? And make you a person with a risk or with no risk of developing the disease. And in case you have the risk, in case you already have the disease, what is the best or what are the best procedures and measures and interventions to treat that disease and other diseases? Okay, so here again, I'm just giving you quickly an example of what we have in our DNAs. We have on the left side chromosome that comes from the father, the right side, the chromosome comes from the mother, and then we have the sequence, for example, the GATCAC. Okay, in both sides is exactly the same in this picture. In that position where we see normal, the circumference, the ball, the green written normal, this is what we expect to have in most of the population. So what means normal? Most of the population are like that. Okay, so when it's look to the right side, if we consider this variation, for example, in the FTO gene, the gene related to obesity, we have a normal person. Phenotype, normal. He's not obese, he's normal. So we have the alleles expected AA, which means homozygous. Homozygous is something which is the same, AA. If we had, in this case, AC would be heterozygous, but we have an AAA. Below, we have A bigger than C, means that most of the population are A, and the smaller part of the population is C. When we talk about smaller part of the population, we call also as the risk factors. So the populations with C instead of A, they would be in a risk. Okay, so let's change to the next slide. And now we can see clearly what I'm saying. This gentleman, he instead of having AA, he has CC. That means from the father, he has a C. From the mother, he also has a C. And that means he's homozygous, but not homozygous of reference, because in this case here, it's a homozygous of reference, meaning that who are AA are most part of the people. It's a reference. In this case here, is the homozygous of risk. So that means he has a change. He has something not expected to have in his DNA sequence, and the phenotype is an obese individual. Okay, and how does it happen? The RNA comes, makes a transcription of the DNA, and after that, you have the translation of the DNA. You're going to have the codons of the amino acids, and the proteins are not going to be good protein. Okay, it's not going to be a perfect protein. And when it happens, you have a change in your phenotype. So this is for everything, for cancers, for inflammation, for neurodegenerative diseases, for everything. When you have something different from the expected, even though sometimes you don't have two of risk, but only one risk, it could represent also a change in the protein or not. Okay, so that's why we call heterozygous when you have the factor of risk, you're a carrier of one allele of risk, in this case coming from the mother. You have the possibility of having a phenotype of risk or not. In this case here, the phenotype of the gentleman is normal, in spite of being a carrier of allele of risk. Okay, well, this is the panel that we call the Men's Health Panel. Okay, so when the patients come to one of our doctors in the world, sometimes they say, "Doctor, I want to know something about my health. I just want to know something about what's going to be with me, what can happen to me, and what I can do to prevent that." Sometimes we have patients that they say, "My mother had Alzheimer, or my father had a cardiovascular disease or a cancer. I want to know if I have the same chances to develop those diseases." Okay, look, genetic is a kind of lottery. It's not because you had a father with Alzheimer or a mother with Alzheimer that means that you are going to have. Okay, it depends on many factors, and our test can show you how far you are of developing those diseases and what's your risk of developing those diseases. Okay, so this is the sample of a man's health. We have on the left side a big map with some diseases that we call the executive panel disease, where we have information about your mind, your brain, your thyroid, your heart, your stomach, pancreas, your gut, intestines, blood, prostate, bladder, kidneys, adrenal gland, liver, lungs, and eyes. This will have the same for women, okay, where we have information about breast cancer, ovary cancer, and also you have other information that belongs to women and men. All this information belongs to them. Okay. On the right side, we have also the radar maps. The radar maps are very important because the radar maps show us the main factors of risk. And if you are homozygous, remember what you saw before, meaning that you are red or you're red green. So if you are red, that means you carry two alleles of risk. Okay, it means that you are going to be in the red map in the red. If you carry normal 0, I mean the normal, you're going to be green. Then you have varieties of results according to what is homozygous, meaning that if you carry one red, one green, meaning one a little risk, another not from risk, you have a median average. And this average is a very good calculation using all the genes involved. For example, in this panel, we have more than 3,000 genes related to men's health. And then we have a calculation that is done regarding the risk of the patient. Okay, so this type of analysis is very precise. It's not the only analysis that we do. I'm going to show you how complete we are. Okay, because this is one type of getting information. We analyze also something very important, especially now that we are working with Asia. It's very important to inform one thing. We do the breakdown of the ethnicity of each patient. Why? Because unfortunately, most of the scientific researches, they were done or in US or in UK or in Germany using Caucasian population. Can we consider this analysis, these studies, these articles, publications done only Caucasians valid for all over the populations in the world? Unfortunately not. We need to do a calculation of risk. I'll give you an example. There are some people that has a mutation called MTHFR 677. Okay, and they have some serious risk of developing a high homocysteine. Okay, which is that it can lead to accumulation of lipids in the artery walls. But except two populations in the world, this is very serious. For two populations, we cannot consider a factor of risk. So we need to understand what is your composition, genetically speaking, to know what type of risk you have, what type of problem that you can develop in your life. Okay, so this is for everything: for cancers, for Alzheimer, for cardiovascular disease, and diabetes. If you belong to other populations that are also studied, we can tell you the level of risk that you have. Okay, it's not the same level of risk for everyone. We are not the same. We are 99.9% the same people, every one of us, but the 0.1% carries more than 6 million genetic variations. And we are different inside our DNAs in terms of predisposition of diseases, in terms of how we respond to foods, how we respond to different medicines. What does it mean? Why some medicine is very effective to someone and not for other people? Why some medicines, some treatment, for example, the treatments for cancer. Today I was talking to a patient. He's starting now a new chemotherapy and asking us, how sure can I be if I'm going to be successful in this chemotherapy? So checking the genetics, we have this information. How effective it's going to be the treatment with methotrexate, with vincristine, for example, cisplatin, and so many other drugs for cancer, and how toxic it's going to be for your body. So this is very precise. This is according to the FDA. This is not something that we created. It's something that we use information. We gather millions and millions of information. Until the end of 2023, our database will reach 30 million scientific articles. So this is very important to determine the coefficient of risk. So this is also a very deep information inside our reports. Okay, I'm gonna show now a little bit about our reports. But before showing our reports, I would like to talk to you about one specific gene. Okay, just reminding that in our system, we work with 26,617 genes. 26617. Okay, one of the genes is the gene called MTHFR. Okay, MTHFR. Why this gene is so important? Why is it so important? Okay, it's not the most studied genes. The most studied gene is the gene TP53, which is related to cancer. But this gene is being spoken in many seminars that I participate all over the world. And let's talk a little bit about it. What does it do? Okay, just to understand, the gene is a position of our DNA, okay, that is responsible for making what? To make a protein. And a protein has a function in our body, okay? There's a function. And the function of the protein MTHFR, as you can see in the bottom left of the screen, left side, MTHFR, this protein is an enzyme. And what's the function of this enzyme? Is to break and transfer a molecule. Okay, so what does it do, the MTHFR? What's the function of that? You know that we eat vegetables, we eat flour, like a wheat flour, bread, pizza. We get something that we call folate or vitamin B9. Okay, folate. The chemical industry produces the acid folic or folic acid. Okay, it's also B9 to do enrichment of the. Okay, we do the enrichment of things that we eat. So every time that we go to a market and we see in the shelves a product, let's say a morning cereal is written enriched with vitamins. We know that enriched with vitamins mean enriched with folic acid. Okay, and folic acid is something that is given to pregnant women all over the world. When a woman is pregnant, goes to obstetrician, and the doctor will say, now you need to take a higher intake of folic acid to prevent neural tube defects in your baby. Okay, great. But one thing that the doctors don't know is that 30% of the population cannot process this folic acid or folate. Okay? Folate comes from vegetable sources, and acid folic comes from chemical product. Okay, that is used for improvements in the food industry. So, what is the function of this enzyme MTHFR? Is to break down the folic acid. And what's to break down the folic acid? It's transforming the MTHFR has the function of transforming the folic acid into 5-methyl THF. Okay, so what's the main function? Is to transform that in the end of the day will enter another pathway that we call the methylation pathway. Remember that we talked in the beginning? We talk about what? About methylation. Let me come back some slides. I'll talk to you about that. Methylation. Remember that? It's the position cytosine receives a CH3 or H3C. You can see on the right side, receives that. And to receive that, it's a process that we call methylation. So this round in the middle, where you have SAM, methionine, is responsible for donating this methyl, okay, this CH3. Okay, so if you do not have the MTHFR working perfectly, that means your methylation pathway, this wheel that must work perfect in order to donate the methyl, is not going to work. So we have a problem. What's the problem? The genes that we want to keep off, they're not going to be off. So when we talk about the genes related to cancer, that our body knows this gene should be off. Your body is not being able to give the methyls to keep it off. So this gene will be turned on. Not only for cancer, for all diseases, for features, for everything. So this pathway that we see in front of us, that we call methylation pathway, is extremely important to work. Okay? And if it's not working, if MTHFR is not converting the folic acid into methylfolate, that is used inside this pathway, what can happen first? Okay, first of all, we are not producing methyl. We're not donating methyl. We're not giving this methyl to our DNA to cancel the genes. Second thing, we're gonna be increasing our homocysteine levels. For those who are cardiologists, you understand that the homocysteine levels, high homocysteine levels that we see here in this drawing as HCY, if they are high, that means that this patient is a big candidate for cardiovascular diseases. Why? Because if you have a high homocysteine, you'll be accumulating fats, lipids in the walls of your arteries. This is very dangerous, okay? Another thing that you cannot see in this drawing, but it also creates a very bad impact in our antioxidant system. Why antioxidants are important? It's not about what you see in the news, the supermarket, oh, important for your health, antioxidants, but let's talk about scientifically about antioxidants. Why they are so important? Because antioxidants, they are good to do many functions of repair in our body. For example, the DNA repair. We need antioxidants to be working. In order to do the autophagy process in our body, what does it mean? To eliminate the old cells, the senescent cells, we need the antioxidant system to be working. So if this methylation pathway is not working, what's going to happen? You're not going to be able to produce the most important antioxidant that we endogenously make. What's endogenously? Inside of us, we make. That is the glutathione. Glutathione comes from this pathway as well and uses the glutamic acid together with the cysteine and the glycine, okay? And requires not only the B6, but also the vitamin B6, but also requires that this pathway to be working perfectly. Otherwise, we're not going to have that. So what we see more and more, people with variation in this MTHFR, they have many susceptibilities to develop diverse type of diseases. So here is only one gene. Imagine that I'm talking about 26,000 genes, each one of them with a function. Okay, so let's talk about this specific genes, the MTHFR, responsible for turning on genes on and off, building and repair DNA and also RNA, processing chemicals and toxins. We know everything we eat is full of toxins. You know, we have also the ammonia problem in our bodies. Processing hormones, okay? Building immune cells, turning the stress response on and off. Energy production, reducing histamine levels, neurological development and function. So everything that we talk about regulation in our body, we depends also on this gene. So if you have a mutation or an alteration, a variation in this gene, it can really impact directly your health. Okay? Many studies, even now, they are connecting MTHFR with autism. Okay, I do not say that until now. Okay, but it may be something that in the next years or even in the next months being brought by many scientific researchers. Okay, why? Because there are some close statistics saying that women with this variation in this gene MTHFR, if they take folic acid during the pregnancy, they are not going to be able to transform the folic acid into methylfolate. If they do not have this transfer, they're going to be accumulating folic acid in their bodies, and folic acid will be transferred in the CI acid, the cis form, which is very toxic. And the cis form can contribute for autism in babies. Okay, so that's what is being studied yet, is not confirmed, but statistically, we can see that it can happen. But what's the suggestion in this case? I suggest for the patients to take, instead of folic acid, take methylfolate, L-methylfolate, because in case that this woman has this variation, she doesn't know that, and this variation is within 30% and more of the population, she's not going to put her baby in risk. Okay, this is one of the things here, because we are short in time. I'm not going to explain a little of that, but depending on the variations you have more and loss expressions of it. Okay, and there are other things related to that. Okay, so now let's talk about exome. I want me to do the Manus cell panel first.
Yeah.
I can go. Okay. So I'm gonna do the before entering here in the exome. I'm gonna show the Manus cell panel. This is our panels. The microbiome. Perfect. Just get the color here. This is for cancer. Okay, this is the Manus Health panel. I'm going to increase my screen a little bit more so that you can better see it. So here we have a patient. Here's the disclaimer, how to interpret the panel. This talks about the statistics. And now we're going to see what the patient has, okay? This is a panel of 52 pages, very, very precise and very well developed to make fast for the doctor to interpret it. Okay, so this is our category. So the first category, second category, and then the other category. The first category is the disease susceptibility of the endocrinological system. So this patient here has homozygous of genes for type 2 diabetes. Okay, also insulin resistance, also for metabolic syndrome, which is a disadjustment in hormones. Okay, here has also some risk, but means that he's a carrier of variants. But here he is AA or TT or CC. That means they are homozygous of risk for these diseases. So the chances of developing diabetes is very high. Okay, so what age I can do the test in my patients? You can do in a baby even. The baby is going to see that. So that gives also the parents the notion that what I can do in order to reduce the risk for my baby when he's older, for example, to develop type 2 diabetes. That's creating good habits, so on, so many things we can do. About endocrinological system and hormones, we have here an individual with higher levels of testosterone. Usually, we don't find high like that, okay? But this is not a balance. The balance should be here in green, but we have sometimes low, which means in blue, or high in red. When we have this in red, means that this patient tends to have more aggressiveness and other conditions related to the higher testosterone levels. Also, this is the patient with a more benefit of melatonin. Imagine that this patient travels a lot and needs to sleep. So he buys melatonin, he doesn't know how much to take. Oh, doctor, I take 3 grams, 5 grams, or 10 grams, milligrams, for example. I have many patients that take 10 milligrams. And I say, no, you have a very big benefit if you take melatonin because your metabolism. So you need to take 1.5 milligrams or 3 milligrams to be more than enough, okay, for you. This patient is a male, so he has, in this case, also a low progesterone and a low DHEAS, okay? And his oxytocin level is normal, okay? Now let's talk about cardiovascular system. Okay, this is a summary of 8 most remarkable conditions according to his DNA. And here we have hypertension, cardiac arrhythmia, high ferritin, atherosclerosis, okay? And then you have some alterations that also are related to cardiovascular diseases, but meaning that he is a carrier, but he's not in a very high risk, okay? Even for LDL cholesterol, he can have a slightly higher triglycerides, but he's a patient for high hypertension, high cardiac arrhythmia, high ferritin levels, and high risk atherosclerosis. If this patient is, for example, 70 years old, the first instruction, let's do a scan, let's do analysis of your carotids to see how is you related to the lipids accumulation in your arteries, okay? And another thing, remember that a few minutes ago we were talking about MTHFR, right? This patient, male, he's a carrier of one variant, 677. He is not full carrier, he doesn't carry two of them, but one. And this one is enough to compromise his methylation pathway. So it's enough to reduce the effectiveness of making the genes to be turned off. See how interesting? We can see that here. Okay, only one gives a reduced effectiveness. Okay, let's go now to the next page, neurological system right here, and we see a patient with very high Parkinson's disease risk, also high stroke risk, and average Alzheimer, average sleep quality, normal mental cognition, cognitive decline with age. So it means that the patient has risk for medium risk for Alzheimer, but very normal risk for cognitive decline with aging. His dopamine synthesis is normal. Serotonin synthesis is normal. In case the serotonin synthesis could be altered, let's say for him here or even low, that should mean that this patient should be doing adjustment in the serotonin production. How we can do that? We know that 90% of our serotonin is produced in our gut and only 10% produced in our brain. So this is a very important neurotransmitter related to also to depression. Okay, many studies today also connect dopamine to depression. But as we know, serotonin is connected to depression. So what we can do? In terms to increase that, we need to give to this patient some right probiotics, fibers, in order to produce more serotonin inside his gut. And also the L-tryptophan and other serotonin precursors, okay, that will be helping on that. So there's another other layer in our system that you're going to see a little later on, how the prescriptions are created, okay? So the same thing for behavioral, the system generates your behavioral based on your genes. And this is in the beginning when some papers came out about behavior and genetics. Myself, I was very skeptic about it. I said, no, really, maybe not, no. But the quantity of papers and quality papers and information, and then when we started to relate them to patients, to real patients, we saw how precise they are. So today, we have even a behavioral panel specific for that. This is only a small pieces of information, but it's very precise. We can detect many characteristics on the individual. And right here, for example, relationship selectivity, difficulty dealing with criticism, anxiety. But this patient has more what? Has more impulsivity. This is what is really showing out here. Okay, impulsivity is what this patient is showing. Fears, normal. Mood disorder, medium for depression, and so on. About gastrointestinal system, this is interesting. Why? Because some people, they make confusion between lactose intolerance and milk allergy. They're different things. Because the lactose intolerance, we're talking about the MCM6 gene, intolerance to the protein, to the sugar of the milk, which is the lactose. And here, the milk allergy is related to the protein of the milk, which is the casein. Okay? So in this patient, what I have more, milk is not good for him. You can see here, he is a carrier of one allele for lactose intolerance, meaning that he can have that, but not so expressed. It's not here in red. But in red is the milk allergy related to the protein of the milk. So this patient is not recommended to have milk or dairy product. Okay? His risk for celiac disease is normal. Gluten intolerance is average, but he's a carrier of inflammation. So what does it mean? For ulcerative colitis, he can develop because he carries one gene. So that's why it's so important to keep his methylation pathway flowing well, to keep the bad genes of inflammation low. But in terms of, say, it's not because he doesn't have the risk for celiac or gluten intolerance, it's also very low, that this patient can abuse the gluten. Because we know gluten is very inflammatory and will create a problem soon or later for this patient in terms of inflammation. Okay. About oncology, we look in here four main genes, okay, four main diseases actually related to oncology. We have also a panel for cancer with dozens of conditions. I'm going to show you later on. But this here shows 4 most common types of cancer, like the colorectal cancer, lung cancer, prostate cancer, and thyroid cancer. This patient here, if he smokes, is a good suggestion to stop smoking because he carries the genes to trigger the lung cancer, also for prostate cancer, and also for thyroid cancer. But for colorectal, he's very average. In this term here, we need to provide him the right diet and also the right antioxidants to fight against these susceptibilities that they have. Okay, and also we need to check some other genes that we do in our system. I'm going to show you later. When we check exactly the positions in charge of making the proteins, okay, and that we call the exons positions of your DNA. Okay, so that's why we're gonna deep into that more precisely to check the levels of risk for this patient. Respiratory system, we have here a patient with asthma, a lot of genetic related allergic rhinitis, but not so much apnea and pulmonary emphysema. So what you need to understand, he's allergic person. Okay, he's allergic person. Osteomuscular system, a lot of risk for rheumatoid arthritis, a lot of risk of osteoporosis, and a medium-high risk for knee arthrosis. So again, we need to prepare to this patient a diet low in inflammatory ingredients. Okay, so bread, milk, sweets, we need to keep out of his diet. We need to reduce in his diet because he has a very high inflammatory risk. Sensory system, macular degeneration with the age. Oh, well, what can I do? You can start from when you are 40 years old, 45 years old, you start taking the right antioxidants, the carotenes, the lutein, and also zeaxanthin to reduce your risk for macular degeneration. Okay, this is also glaucoma. He's very mild. And then we go to the map that we saw before in the PowerPoint. It's a map from one patient, okay, where we can see some other risks that the patient has. Okay, so this one is interesting. That's what I want to bring to you, because this patient carries the variation for atrial fibrillation. Okay, specifically in this case, he's a twin brother, and his twin brother passed away from atrial fibrillation while he was training in a sport gym. Okay, so here we see the atrial fibrillation very high in homozygous in red, and this is a silent disease because most people carrying atrial fibrillation, they cannot detect in routine heart checkup. Okay, sometimes we are lucky we can find, but not always. So that's why it's important to see that he carries this mutation for atrial fibrillation and also for cardiac disease, also for hypertension. Okay, so this is our some information we see about his pancreas, about insulin resistance, type 2 diabetes, and for everything we can do. In this endocrinological system, we have some information which is very relevant for each one of the diseases. Here, the endocrinological system is very important to understand. We have the same category that we mentioned, we have the metabolic syndrome. He's a carrier of plus. Remember the first slide that I showed, plus meaning that he carries from the father and from the mother the risk. Okay, so 1 full variation for metabolic syndrome, 7 full. You see here, 7 for type 2 diabetes. Today, I had a patient with 14 variations for type 2 diabetes. So it's a very urgent case to treat with pre-diabetes. So this patient we can see already here. So for all the other diseases, we can see conditions, not the diseases only, we can see the variations. In this case here, the melatonin is a benefit as 1, but for progesterone, he has a tendency for low progesterone because he carries 2 variations related to low progesterone levels. The cardiocirculatory system is the same. See here, you have even here other variations and mutations. You have even the DGFR. And here we have, for example, 13 heterozygous for triglycerides but not 13 homozygous. If it was 13 here, this would be red. But he has red for atherosclerosis, red for hypertension. That means he has a tree which is strong enough to give him a red color because there are 3 homozygous ovaries. The same thing for neurological system. We see here high for Parkinson and high for stroke. Okay, for Alzheimer, he has only 1 variation, which is not related to very high risk, and 11 related to heterozygosity, but it's not the APOE4, which is the most serious variation to Alzheimer. Okay, so this is the first part, and then we continue to see. Yeah, yeah. So we're going down to see the genes involved in each one of the conditions that we show. What are the genes involved? For example, given this result of the low level of the HBS, you have the analysis of all these genes. Okay, to give this score, for example, of testosterone, we have these genes here. Okay. To have the information, let's go to another one just to show you. Atherosclerosis, we have these genes. Okay. For each one, each information, for the mutant one, hypertension, we need to analyze all these genes. So when you get some tests, especially the recreational tests, I say that they are very dangerous because they don't give you a full interpretation and the full analysis. They can just predict how you have a hypertension risk, analyzing, for example, the BDNF. Okay, this is not only this gene is related with the BDNF even to your behavioral. So it's a combination of factors, combination of publications, combination of articles, and consider each one which a certain score of risk here that will attribute to a high risk. It's not only based on one gene. We cannot predict or even inform things based only in one gene. And some exception of the TP53 when you have a mutation is another thing, or the BRCAs, but usually we cannot. Okay, so just telling you the quantity of information that we analyze. In this panel, we deal with more than 3,000 genes. Okay, so this is very important to show. I would like to explain about the panel of diabetes. It's okay to show now? Okay. Okay, so let's go directly to the cellular level that we call the diabetes panel, and I selected some samples to show you. So just getting the diabetes, many issues. Diabetes, and let me get another one. This is another patient. Okay, so we have here 2 patients of diabetes. Okay, that's the first one. It's a man, also just generated today. Let's go directly to have also this maps, but let's go to the cellular level. So I'm going to show you X-ray of the cell. So here you can understand one patient. Let me just reduce a little bit so you can see. It's one patient cellular level. Okay, so what we have here, we have in yellow the insulin that is produced by our pancreas that goes into the insulin receptor normally. You see here, there's no traffic, it's just normal, just go inside, and then comes to the RS1 transporter that carries the insulin to the middle of the cell where it activates the GLUT4 translocation. GLUT4 is a receptor for glucose. And the glucose that we have in our blood enters the GLUT4, okay? And then goes inside our cells. Then we have all the Krebs cycle in the mitochondria to generate the ATP, okay? So what we see here, there is a problem. Here, there's no traffic. You can see enters normally, but in the RS1, you see the insulin, they are bent a little bit. See outside here. So that means the insulin is not being transported properly to inside the cell. If it's not transported to here to do the signaling information to the GLUT4 that is here inside the cell to go outside to the membrane of the cell, we do not have enough GLUT4s around the cell. Means that there's not enough entry doors for the GLUT4, for glucose to enter the cell. So that generates accumulation. You see here, 3 stopped. See that? 3 stopped here. And a lot of G's here inside the cell. Okay? If we see the translation of that, we see normal, 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 and medial. But no, why GLUT4 is normal? Because the function of the GLUT4 is normal. The problem is the signaling. And here, for example, here in Middle East, most of the patients that we find here, they carry this variation. Okay, so the problem that they have is not about GLUT4. The problem they have is not about the insulin receptor, not about insulin resistance. It's about the transport. Okay, so now let's look at another map. Let's look at the same panel, diabetes, but for another patient, so that you can compare the difference. Let's go directly to the cell map. And you can see the difference. Oh, you see here? Remember the other one where you have no traffic to enter the cell. You see here the insulin, no traffic. This one is a big confusion. See that? Big confusion. You have the insulin causing the traffic here. We have these white balls. What is this white balls? These are the lipids, triglycerides. They are disrupting the entrance of your insulin receptor. You see that? So the other one you don't have. It's okay. The fat molecules, they are okay. They are right here. But this one here, they are too much. And they are creating a traffic problem to allow also the insulin to enter. Second problem detected. Again, the insulin receptor, the insulin transporter, EIRS1, are not working, meaning that not gonna put enough GLUT4s outside the cell. Okay. Second thing, you see a lot of traffic here. Again, different from the other patient. Only 3. You have another one with a lot. Why? Because the fat's also accumulating in the insulin receptor and also accumulating in the GLUT4 receptor. So that prevents the GLUT4 to work properly. Means that the glucose is not entering here. And what can cause in the patient? Look, accumulation of glucose in the blood, accumulation of insulin in the blood. That will signal with the time the pancreas to develop the chronic diabetes. So it becomes so much insulin, so much glucose, and the glucose will be different from this patient here. See the glucose alone. Here, glucose alone. You see the hemoglobins? The hemoglobins are empty. The other one here, we see the hemoglobin reached with the glucose, and we call HB1. You see the glycated hemoglobin. This is the glycation process. What is the glycation? It's when you have your cells attacked by the sugar, attacked by glucose, for example. And which cells are the preferred ones? The hemoglobins, the joint cells, we call the collagen cells, also for the skin. So when we find a woman with this problem, we can make sure that her skin will be very aged. She's gonna have a quicker degeneration in her joints because of the collagen degradation caused by the glycation process. So it's a lot of things that we can detect. But doctor, what you can do to prevent it? Okay, first of all, we see that the KPN10, that look here in this drawing is even funny. It looks like a gun, but the function is exactly the gun is to destroy the fat's molecules here to avoid them to disrupt the entrance of insulin and glucose. In this case, it's not working. In the other man, you see that it's working. This one here is not working. So let's see the summary. First, KPN10 high alteration, not working. So you have a higher concentration of fats disrupting the entrances. ANPP1, which is this entrance here, works more or less, but you see that what happens? This is medium. It's not bad at all. But because of the fats, is not working. What we need to do to solve it? To reduce LDL and triglycerides level in this patient, because the fats are really causing a problem and leads to a type 2 diabetes. Second problem about GLUT4, normal. You see, it's a beautiful receptor. It's working perfectly, but it's not performing the right function because the accumulation of lipids in the cells. Okay, and the other thing is the RS1, which is the transporter, is not working. Okay, so as this is not working properly, I need to put more GLUT4s around the membrane. And what's the best way today, known today, to put more receptors outside the membrane? Who knows that? I can tell you, you can find in many literatures, in many articles, and many endocrinologists also recommend that, is to do the walking 30 to 35 minutes a day. When you do 35 minutes a day, and I can tell you that I tested myself, okay, I have myself, okay, I have all the 4 here, I have high. So I'm a big candidate for diabetes. My father diabetes, my mother diabetes, I didn't develop that because I found these issues about the inside the cell that we can see in our test 5 years ago when I started taking care of me. The first thing that I did, walk 35 minutes a day. When we walk 35 minutes a day, and when we put an Abbott sensor in your arm, you can see that very clear that your glucose level, even though that is not more than 100, goes down because you translocate GLUT4s, the receptor for glucose, from inside the cell to the membrane. So if I put more, I'm going to clean my blood from glucose. Okay? Because what's the function of the insulin? The insulin enters here, and then the function is to be carried here to inform the GLUT4, which is inside the cell, to go out. It's just a signaling. Insulin mission is to signal, give a signal to clean the blood from sugar. Okay, so it's not everybody, you are going to be the diabetes because you have a problem in the beta cells that's inside your inlets of Langerhans, inside your pancreas, that are not going to produce the insulin. It's not always like that. Oh, you have an insulin resistance, which is a resistance here. Always, not always like that. This patient, for example, doesn't have insulin resistance. It doesn't have. This problem is about the accumulation of fats. Okay, so this is another panel that we can see. And now we're going to see the oncology panel. Okay, let's talk about, let's go directly into a cancer panel. Okay, so now we're going to see, that's another real case. He's a 40-year-old patient. Okay. So this patient show here that she has many risks. Okay, this is a real case. It's a patient that has cancer in the brain, and this patient, she presented, seen in our test here, many different types of cancer. What really worried us is that because her metastasis level very high, you can see here, it's also genetic. So her metastasis is very high. So the general risk for neoplasm, which is related to hereditary cancer susceptibility syndrome, also very high. Lung cancer, because she's young, she's 40 years old, this is not calling its attention at this moment. Cervical also no. Glioma, yes. Multiple myeloma, yes. And colorectal cancer, not in this age. But we found some issues about cancer, which is not common to, okay? For example, most of the patients we find like here for cancer, like normal. But here we saw some alterations. So even the breast cancer specific panel, we have one only for breast cancer. We analyze different types of breast cancer. For example, if ER positive or progesterone positive, HR positive, or the markers for breast cancer. And what's is nice is that our test doesn't check only the BRCA1, BRCA2. You know, Angelina Jolie checked the BRCA1 and found a mutation and decided to do the mastectomy. Okay, this is the instruction even from the cancer.gov website. But what happens to many women they check the BRCA1, BRCA2 genes, which are the most studied genes for breast cancer, but not only the exclusive one. There are 178 genes for breast cancer today. Okay, BRCA1, BRCA2 are only 2 of them. So all genes need to be studied. Out 178 to know your risk. So for many women who are negative in BRCA1, BRCA2, they could be positive in other genes. So this is very important to test with us because we do the full. That's also one of the reasons we call full of all the other genes related to breast cancer. Yeah, I'm going to show a target exome as well in continuation. I just want to show you the chemotherapy effectiveness. Okay, for these patients, it's a normal chemotherapy response, which is good, not low. Allogenic rejection in terms of organ transplant, normal. This is also normal. About genetic diseases, she has no genetic diseases because if she has a mutation, it's not a disease in this case. It's a genetic disease that can lead to a cancer. Okay, but it's not. She doesn't have. Summary of results. What we can see here, we can see here colorectal cancer, multiple myeloma, glioma, cervical cancer, lung, the neoplasm, general risk, which is hereditary cancer, metastasis high. The rest are medium high, but she also carry homozygous, okay, for many other issues, okay? And let's go until we find the Fromeni syndrome. Okay, let's go to here, where this patient carries one mutation here, which means coming from the father or the mother, or this is what we call this in her case could come from the mother. It's the somatic or germinative, or called the germline. Okay, because not always they say because I have a susceptibility because my father or because my mother. No, you can have something we call de novo or germline that you develop during your embryo stage. Okay, and in this case of this patient, I'm going to show you what we found. Okay. I'm going to show you her results, and this is all the cancers that our test analyzed. Okay, this is very, very, very precise, and give us instructions to go deeper in the analysis. So, for example, now I found that she has all this risk of cancers, and her oncologist sent to us this. Okay, let me increase the page. Just hiding the name of the lab, just because it's not fair, to show the name. But this is a result from January, saying the following. Okay, this is how the genetic exome results comes. Okay, from many people. So for most of the doctors, they don't know how to interpret that. They spend hours, and my God, in many cases they come here non-conclusive, especially for breast cancer. But what do we see here? One pathogenic variant identified in the gene TP53. Which is associated with autosomal dominant Li-Fraumeni syndrome. Remember that I just saw this patient was yellow, saying it's a medium risk, but she has one variant from or the father or the mother or germinative. Okay, it's heterozygous. What means heterozygous? You can see here heterozygous. Heterozygous means it's not the same letter. You have one plus, one negative. In the case, the variation C and D, the patient is or G and A, or the patient is really C and D, depending on the version of the analysis. Okay, so TP53, what is this gene? This is the most studied gene in the world. Okay, so when you ask me, what the gene that the professors, the scientists, they study more, they study TP53 because this is the gene that controls how your body will deal with the old cells. You know that we make, every one of us, every single human, we make more than 1,000 or 2,000 cancer cells a day. Okay, but even if you're healthy, say, I'm a doctor, I do sports every day, I eat healthy foods, even though your body is producing from 1,000 to 2,000 cancer cells. And your body will deal with these cells different ways, depending on many genes. One of the genes that regulate what we're going to do with the cells is the gene TP53. Why? Because the genes give instruction to our body to clean the cells out, to clean it, wash it out of your body. If you have a change in this gene, TP53, means that this gene is not working well. So the instruction of destroying these cancer cells, of eliminating them, is not going to be effective. If it's not effective, what happens? You'll be accumulating cancer cells, and with this accumulation, you develop a tumor. Okay, so that's what mutation means. There's a mutation. She is heterozygous for this mutation, which is pathogenic. Okay, great. But the lab delivered this result. Okay, so detecting the Li-Fraumeni syndrome, which is a syndrome that is very rare, and it's a syndrome that triggers many types of cancer, one after the other, especially in people more than 4 years old. So this is a big surprise in a child of 4 years old. Okay, so this laboratory analyzed 56 genes. Okay, 56 genes only. Comparing to us, I like to show, not to compare talking bad about other laboratories. No. I just show why it's so important to analyze many genes. We analyze only for cancer 8,492 genes, as you can see at the left side. Okay, instead of analyzing 56 genes, we analyze 8,492 only for cancer. Okay, so this is very important because we covered much more than any laboratory in the world. This is the price of the lab. I just splitting that. What we found, okay, we ran our exome. Okay, we ran our analysis of the exons. What is exons? Exome. We have our DNA. The DNA are divided in parts. Okay, and these parts of DNA we call the genes. The genes. Genes has some divisions inside the gene. They have some divisions. We have an area of the gene that we call exons, and we have an area that we call introns. Exons and introns. Exons is an area that corresponds to less than 2% of our DNA. It's just a small area. It's very limited, less than 2%. Okay? And the other area is what we call introns. We decided to focus on both of them. We analyze exons and we analyze introns, and we analyze both of them together. The laboratories in the world, they analyze only this less than 2%. Okay. Why? Because the exon gives information how to make proteins, how to code for proteins, how we can determine a diagnostic, for example, through the DNA, we analyze the exons. But the introns, which is the other part of the DNA, is also very important. Why? Because they regulate the function of our body and also the function of our transcription, of how the DNA is read for everything. So we need to do a full analysis in our genes. Okay? So let's talk again about this patient. Let's go back. As you can see here, one mutation found, gene TP53. We ran the same formation of the patient. We collect the DNA and we collect through the saliva. We don't need the blood to collect. We collect through the saliva. We get specific details in the cell of the leukocyte in the mouth. And look what we found. We found 6 mutations related to cancer. Actually, she has 43 mutations. Okay, this patient is not common. Okay. Usually, I find in patients like 3 to 7, 8 mutations, okay, and not so serious, related to calcium or to the body mass index. But this patient, I found many mutations, 43 mutations, and 6 of them related to cancers. And the last one, as you can see by alphabetic order, TP53 is the same one that they found. But what is interesting is that I found one mutation related to Li-Fraumeni syndrome, which is also related to hereditary predisposition of cancer. And I found other 5 that you can see above related to the cancer risk, but the type of cancer risk, hereditary cancer predisposition syndrome. Okay, so that what we can tell you, when you provide a diagnostic of a syndrome, sometimes can be have much more information, can be much more precise. So in this case here, can we consider Li-Fraumeni syndrome? We can. Okay, there's not so much difference in the treatment. The patient needs to go to chemotherapy, but we see differences in the way of having diagnostic. So our test is much more comprehensive. And I found other mutations that are also important to control in the patient, maybe not now, but later on the line. Okay, she has some collagen-related mutations that are not so common to see on people. Okay, that it's important to control that in the future. And also the breast cancer, she carries one BRCA1 and two mutations for BRCA2. Okay, so this is also other complication that we can see. Okay, about this is what I said. The competitors just carry one pathogenic mutation. We carry for the same patient 6 pathogenic mutation related to cancer. Okay, this now I'm going to show you just splitting the screen. I'm going to show you our genetics, what we do with this data. Okay, just a second. MagGeneCare. So this is the MagGeneCare. This is a system that also belongs to us. What's important is that to explain to you, all the systems that we have, all the platforms, all the artificial intelligence, all the apps, they're developed in-house for us. Okay, they're developed by ourselves. We are not in the hand of any third company. We don't want to stay waiting for them to give an update of information from the genetic databases, from the publications, and have to pay monthly. No, we develop in-house everything that we need. So that's why we are very accurate and precise, and we know what we're offering. That's why our company, we do not do marketing. In spite of, I think marketing is wonderful to promote, to explain, and so on. But up to now, we have more than 5,000 medical doctors just mouth to mouth, one telling about us to the other. And this is working very nice. Well, this is the MagGeneCare system. Okay, this is MagGeneCare. This is your first contact with them. It should look like a little complicated, but it actually looks like a website. What does it do? Well, I'm going to select one patient in my patient list. Okay, so I have here some patients. I'm going to have one of them. Let me get I got one here. That works with this. Okay, I got a patient of 51 years old. Okay, he's 76 kilograms, height 172, body mass index of 25. So he's a little bit overweight. Okay, so I have a list of my patients, the exams of the patient, the diseases, the drugs, supplements, and foods. First thing I'm going to do, I'm going to check his health conditions based on his genetics. So I click here in set results, I go to genetic results, and here I can see what's going on with this patient. The patient reported to me, look, doc, I have a high LDL, high triglyceride. My sugar levels are high. I'm taking metformin and so on. Okay, so what we do here, we have information about the patient, genetic information about him. So the same thing that we have in men's health or in the heart, cardiovascular health, we have here information about the patient. Okay. So I see here very high tendency for triglycerides in terms of venous thrombosis and clots, no, cardiorhythmia, no, heart disease is okay. He carries one heterozygote mutation here, high risk of stroke, so on. Okay, so there are many interesting things. This information is inside the system to be used by our artificial intelligence components. Okay, so what do we do next? We come here to patient data. And we have the name of the patient, the date of birth, everything. And I ask, do you drink? Yes or no? Do you smoke? No. Do you make intermittent fasting? No. And the patients can answer me more things like how you deal with your life problems. Easily, very worried, normal. The patient can reply to me. If you have trauma and other information about high blood pressure, no. High cholesterol, no. Diabetes, no. Have had cancer, no. But talking to the patient and receiving his blood test, I saw that he has a high cholesterol. Talking to him and see that his HbA1c is very altered, he also carries diabetes. Okay, he didn't have cancer, no heart disease, sleep quality is average, fatigue, and so on. Okay, so what I'm going to do, I'm also writing, asking him, do you drink milk? No. List of foods that he doesn't like or intolerant. He said, I don't like food high in yeast, dairy product, red meat, only occasional. I don't like coffee, caffeine, and no take dairy. And genetically, he's not lactose intolerant, but he's a milk protein intolerant, as we saw in the other sample patient. Do you prefer any special diet cuisine? Not heavy food, not so spicy, I want mild. Include selenium, because I saw in the test that he has a risk for prostate cancer. So it's very important to include selenium in his diet. Also, because of some risk that he has for the eyes degeneration, also we consider important that. Okay, but all this information will be used for the composition of his diet. It's not for his composition of supplementation. I'll tell you why. Because our system makes the diet properly for the patient. For example, if the patient said, I want Malaysian food, or I want the South Indian food. I like, let's say, Singapore style food cuisine. The system, our system, prepares the meal according to what you like. We don't want you to be adapted to another diet. We want a diet to be adapted to you. So this is another difference, because how can people eat something that they don't like? I'm going to give, for example, a Chinese food for a person in Mexico. Maybe he doesn't like. So the opposite something also happens. So we need to be very specific on things that you like. And I want to bring all these supplements here, all these minerals and vitamins to the meal of the patient naturally. So we prepare the diet according to what the patient needs. Okay? So also we see the patient needs that. But let's see now other things. The patient also did some blood tests. And when he brings to us the blood test and our system receives information, we can see some alterations. Okay? The first time he did, his HbA1c was high. Now he's controlling. For me, it's optimal. Okay? Yeah, the results are automatically generated. This is very important. Let's say, for example, he's doing again HbA1c. We delete here and we include automatically the blood test or image test. Everything is right here for the test to be generated. Okay. Another important thing is that I can add more disease. The patient had informed this disease. He needs to do prevention of Alzheimer, cholesterol level high, colorectal cancer prevention. He showed me lipoedema. He has this mutation about the folic acid, serotonin synthesis he needs to increase, and triglyceride levels are high. Okay, but I can also add more things. I have here many diseases that I can add according to what the patient has. Okay, just add here. So what's to do with this information for me? Now I click here in diagnostic, and I have all the informed diseases, genetic information, what is confirmed. Okay, let's say what's confirmed based on his test, based on look at that. LDL is confirmed above reference. Okay, the reference minimal, reference max, optimal max, and the patient has 139. So it's above the reference. Okay, and here I have the supplements for treatment, and here I have the foods for treatment for the patient. Okay, here for HCl, for everything that the system, according to his blood test, could confirm. Even his TSH is above the reference. Okay, so okay, good. What I do with this information? Diagnostic report. I come here in crossing. In the crossing, the system gives to me the necessary diagnostic exams. Okay, what I do if I have a high risk of Alzheimer's? What are the tests I can do today? So I have here the list. I have here for the colorectal prevention. What I need to do is 51 years old, you need to do FOBT. Lipodema, there's nothing to do, only a checking manually, clinical checking. For the serotonin synthesis, also no, but for MTHFR, we can check homocysteine, folic acid levels, and vitamin B12. Okay, well, also genetic conditions, not informed by human, also genetics. We have here all the list of exams that we need to take. And this is generated automatically from the report that you saw from the Men's Health report. Okay, so this is what is
not given to the patient.
so the report is not given to the patient. So the information keeps here for the doctor. Okay, so okay, good. But what I do now? Great. Click here in recommendation. And here I already recorded a recommendation for the patient. Okay, but I can generate new recommendations. Okay, and for example, let's say the patient didn't only informed me some conditions. And I want to cross the conditions he informed, but I didn't receive the exam test. So the system is generating this recommendation. Okay, or I want to use only the genetic test results. I can also use that and generate results based on the genetic test. But that's not generated already. Let's open this recommendation. So here in the recommendation is divided all the necessary supplements that this patient needs in his life. Okay, based on his genetics and everything. I will decide what I will recommend to this patient and when he's going to take. For example, acetyl-L-carnitine. Okay, he needs to take in different quarters of the year. The year has 4 quarters. Okay, so the system we determine it is taking the first quarter, second, third, and fourth. Why are we not putting in all of them? Because some supplements we need to give like 3 in 3 months and stop 1 month and start again. We cannot give all the year long. This is very important. They do not have efficacy. Okay, so let's do one for example. Amylase I'm not going to give to him now, but let's say ashwagandha. Okay, he's having an average sleeping quality. I'm going to give him, for example, 100 milligrams of ashwagandha in the first month and in the fourth quarter. So I save it here. Okay. So this is why I want to show you that also many things are recommended, the probiotics and so on. And after that, a list is generated per quarter. Okay, so we have here is also the food, what he needs to avoid, to include in his diet. Okay, and why he needs to have that. Let's drink my coffee because you have an altered ALT index up. Drink more water because you have a uric acid alteration. Drink plenty of water because it has that. So that you have eggs, alterations in AST, which is a liver marker. Okay, but we have here the information. Let's see here by quarter. As I said, I didn't finish his analysis yet, but I can make like the first quarter, then I go to the second quarter. Yeah, so I go to the next quarter. So because we don't have too much minutes more left, I want to finalize this analysis here, and I'm going to open for questions. Okay, and also this lets me know that we have the recommendations here. We have the life change, the suspected diseases,
PGX.
and PGX. Next month, we're going to be releasing the PGX, which is that pharmacogenetics. It's how you work, how your body works according to the drugs. Okay, if you have toxicity and not, and we put in all the information here in our system. Okay, so that's all. I'm opening here for questions now. Thank you so much.
Thank you, Dr. Roberto. very insightful for our doctors. I think we are open for questions right now.
Before we go to the questions, I would like to inform
Okay.
that we do not offer this clinical reports directly to patients or consumers. According to WHO compliance, we need to have a registered medical practitioner, and that's why we always go through hospitals and clinics. And when we are dealing with oncologists and cardiovascular or level 3 issues, we have the specialist who will be doing the genetic report interpretation. So that's how we work. And the reports that you saw is not actually given directly to the patient. So because of the information that are present, you will sometimes alarm the patient and you might panic the patient. So that we leave the doctor to take the call on what to be communicated, depending on the condition of the patient. And also when you saw the MedGene Care, the system is completely AI driven, but still the doctor has the overriding capability of what are the recommendations that needs to be released. So I thought I will include that in the communication. Thank you. And we're looking forward to hear any questions that you may want. You can post it on the chat screen or you can speak to us now.
Yeah, I think that's both ways. Either the chat screen or can ask the questions directly. Okay, while we wait for any questions, when will the doctors who are going to be counselors for this will be ready to offer their services at what stage? Do we need to have more training and then they need to get more familiar with that application, MedGene Care? How does this work?
We will provide a training on the user training on the MedGene Care application. And the MedGene Care is basically designed to intuitively guide the doctors on the whole process. So the report that we provide is more of a document that is kept in hand for a deep dive when they want to know what is the cause of the susceptibility, why is this particular disease expressed as the highest score. If they want a deep dive, the report will be always handy for them. But more than anything, the MedGene Care is more than sufficient for the doctor to directly, precisely come up with the recommendations that he needs to provide to the patient. Because the major focus of MedGene Care is to provide the solutions for the identified conditions.
So it's basically very customized to the genetic results of that particular patient.
Not just the genetic report, but also the blood reports that
blood
the
reports.
patient will be providing. So combining the genetic conditions as well as the blood results and the lifestyle questionnaire, the system generates the recommendations. And that is, you will find it for the food, lifestyle, and supplementations. But when it comes to the drugs, the medicines, our system is currently loaded with all the US and EU approved drugs. But in every country that we are launching, we also load the approved drugs of that particular country as well. So whatever pharmacogenetics will provide is, depending on the condition that is confirmed, we will be able to pick up the medications that are available, the drugs available for that condition. And we will be able to tell the toxicity and efficacy of each drug available for that condition. So the physician can recommend the appropriate medicine which has more efficacy and less toxicity.
Okay, that's excellent.
Right here, a question. To kickstart, should we communicate with?
Okay. To kickstart, Mohan is our country representative, so you can communicate Mohan. Mohan will be able to coordinate the next step. The way how we take it forward is we will provide you with the data room with sample reports. We will try to understand what practice you are, what is the area of practice that you are keen, and what is the areas that you are trying to serve. So appropriately, we will provide you with the sample panels for you to go, and you will also have one more training session directly for the signed doctors, and our team will also provide you the user training for the Medgene Care. So once you have both of that, then you will also sign a commercial agreement, which will all be handled by Mohan. And the test kits that we will present to you, it's just a buccal swab. So you have to provide your patient with a buccal swab. So they do the sample and then they give it back to us. And it usually takes to 2 weeks time for us to come back to you with all the results. And it will be uploaded into your login, the physician portal that you just saw, and your patient details will be in it. So from there, you can access the reports, you can download the reports, and you can provide the recommendations to your patient. And the most important point is it's not just one time. You can, depending on the condition of the patient, you can track. The system can recalibrate the recommendations based on the blood reports if the patient is going to do once in 3 months or once in 6 months to understand how he's been managing himself. So it is, the follow-up is also there in the system.
So rather, if the patient undergoes further blood test, the results from the blood test, how does that get into the system? Who enters the results?
Right now, the doctor or the nurse in charge of the doctor, they can key in the report manually inside the system. But we are also parallelly developing the system where they should be able to simply upload the blood reports into the system and it automatically will calculate. But that will take about 2-3 months time. But right now it is more manual entry by line by line. But the results will be automatically generated, whether below optimal or between the reference points will be automatically calculated.
Okay. So this is what you call actionable diagnosis?
Yeah, actionable diagnostics and solutions. And we will be also very soon, I think probably within the next 2-3 weeks, we'll be releasing one more line item to this, which will also generate automatically the diet or the food recommendations based on the genetics of this person, of the member who have done that test. So it takes into calculation the nutrigenetics aspects of the member, and then it will suggest the diet. So the diets can be automatically generated out of Medgene Care.
Right.
For bigger cases like oncology or chronic critical issues, we do targeted exome, which is slightly deep dive, and that requires the concerned doctor who's dealing with them to provide other informations that they may be having, the patient may be having. So that is a complete report that is generated right now, not within the Medgene Care, but it is handled separately.
So
things like providing chemo protocols, because we will be able to predict the effect of the chemo on the body based on the genetic predisposition, and we advise the oncologists on the protocols that could be more conducive for that patient. So those things are handled out of Medgene Care right now.
And who decides what panel, because you have so many panels, right? So when the doctor consults with the potential patient or client, how does he or she decides, because the patient or the client may not be very familiar or comfortable to decide on, so how does that?
So how it usually happens is usually the doctors will be engaging patients for a particular condition, whether it's diabetic, whether it's cardiovascular. Then the doctor will, because the doctor has been handling the patient for a few months or years, they may want to have a deeper clarity on the causes and things. So in those cases, the panels will be directly ordered based on the critical conditions that the patient is facing. That is one way of generating the prescription for the patient. But if for preventive situations, it's usually the men's health or the women's health, which is more of a genetic screening, a whole body genetic screening is what we do. And once if the patient has any pre-existing conditions, the patient may opt for maybe the patient is a diabetic, so he may still want to try the preventive ones as well as for a diabetic panel. So the doctor might prescribe two panels, women's health and the diabetic. And in certain cases, now the recent number of cases that we are receiving more is on fertility. So the fertility panel is something separate. So the doctor handling the couple will directly prescribe a fertility panel. So depending on the cases that the doctors are dealing, they will be prescribing. Otherwise, usually preventive is through men's health and women's health. And I have another question: is there another training for targeted exomes? Yes, targeted exomes is something slightly deeper. It's a deep dive. So that's a separate one where probably we can work and schedule another session for it. I think we have 38,000. How many genes we are targeting for targeted exome?
For targeted exome, we analyzed 26,000 genes, and the variations we analyzed to 140,000 variations.
Yeah. So targeted exome is very specific. It's more to do with syndromes and very critical mutations. So we analyze the genes, we analyze the 26,000, but in terms of variations, we analyze about 240,000. But that's a separate one. But we will, as requested, we will plan for a separate training on that.
Maybe Radha probably can also tell the participants about how whole DNA, the clinical aspect differs from the other genetic screening products in the market.
Yeah, one of the key things is the number of genes and conditions analyzed. So as Dr. Roberto was saying, for breast cancer, majority of the competitors in the world, they will analyze probably less than 10 genes that is responsible. And we do about 178 genes for the same breast cancer. So the quantity that we analyze is very high. And as Dr. Roberto was saying, that we hold one of the world's largest genomic data. The accuracy level is very high, and the spectrum of data that we use to analyze and produce the report is very high. To highlight a simple factor, we currently hold about 25 million scientific publications in our genomic universe, which is our big data. And whenever we keep saying we have 26,617 human genes, that is the highest in the world. You can even Google, you can chat GPT anywhere, you will find that it is between 22,000 to 25,000. Even Google has only 25,000. Human Genome Project also has only 25,000, but we have 26,617 genes, and it's auditable. So we work with the governments, we work with large research projects. So we cannot say these numbers just like this. So we are now collaborating with few governments on cancer research. So because of this data depth that we have, and one of the things that Dr. Roberto was trying to address, but I think he could, there are a few more things that I would like to add to that, is ethnicity precision breakdown. Especially after 2015, we found a lot of Asian countries going big in genomic research. And that gave a lot of information about different ethnicities, and not limited to the Caucasians or the American research inputs. So to give you an idea, 2021, China surpassed in terms of scientific publications on genomics than Europe and US. They published about 600,000 scientific publications came out of just China in 2021, and India followed. India is doing more than 100,000 every year now. So the data that is pertaining to the ethnicity of various countries pulled in, and that's why we wrote our algorithm, which is ethnicity precision breakdown algorithm. So, for example, very relevant example that I can give is Dr. Roberto. He may look very German. Yes, his mom is German. He's more German, but he also carries a 10% Han Chinese. So the coefficient of the the mutations effect of that particular ethnicity is applied to generate the susceptibilities. So without which many African ethnicity populations may be immune to a lot of variants which the Caucasians may not. So that has to be taken into account and we apply. And we found that we have now millions of articles just on Asia. So that is why we are good to say that it's global. It's a global template now, and not any population specific template that we have. And we can share with you after this call. We can share with you sample reports, and also we will share with you the number of genes and conditions that we analyze for every panel. And that is the key that we need to look for when somebody is offering a genetic report. How many genes are they analyzing, and how many conditions are they analyzing? Very simple example, it's a multi-billion-dollar US entity that's very big in cancer analysis, cancer panel analysis. We even showed that one of the report that we showed today is of theirs. So they have been analyzing how many genes, like 246 for
gene.
okay.
They analyze 56.
So they analyzed 56 gene. So, but we analyzed 8,400 genes. So the value of the result that comes out is very well validated. That's what we're trying to emphasize.
Do we have any? I think while we wait for any questions, we are almost reaching the time. I will share my email here. For those who have not shared your contacts through the Google form, please do if you want to receive more information from us and updates on the results and also the next meeting if you're interested to be part of the panel. And I would probably share more documents which I would receive from Full DNA, and especially on their progress and all the new projects and some of the possibilities that's going to happen in the region, especially in Singapore and Malaysia.
We are happy to tell that we have started our operations in Malaysia, and we have signed with healthcare entities in Malaysia as well. So in fact, some of the doctors of the entity is currently present among with us, and we are looking forward to drive much bigger and expand faster in Southeast Asia.
That's good, because I think this is something we are looking at the future of healthcare and how we're going to drive, healthcare more towards precision health, precision medicine, and I think that's the way forward. And this product enables the doctor, the clinician, to be engaged with your patients, not one time, but on a continuous basis, and to manage their conditions. And especially one of the panels which we have discussed with Full DNA is looking at the conditions in Malaysia, especially the NCDs, and probably we will customize a specific panel for the Malaysian population to tackle this problem, which is almost takes up 70% of our Malaysian budget.
Perfect. And I think it's time. Thank you.
Yeah.
I would like to thank everyone for being and giving their attention. So your support and cooperation is greatly appreciated. We'll keep you posted on the following seminars that we will have. So we look forward to working with you all soon. Thank you, Mohan, and thanks to the great team.
Thank you, everyone. I'm just sending also here in the
email address of Dr.
email also here for those who want to contact any question. I'll be more than happy to answer you.
Any technical queries that you may have, feel free to send the email, and
also,
we'll be happy to communicate back to you.
And also for
Sure.
Radha, who wants to contact, this is Radha's email.
Yes. Thank you. Thank you so much, Dr. Roberto and Radha. Thank you. Thank you, everyone. Good night.
Thank you very much.
Thank you.
Bye-bye.
Bye.